Welcome to class. For those of you watching on YouTube, this is a bonus lecture for my first semester biology class at Gordon College. And Gordon this week just switched a couple of days ago to fully online. It's just before Thanksgiving 2020, and I can see a lot of the students are watching from home already, and a lot of them will be watching on Zoom at a later time. So for those of you who are live here, thank you for coming. I know you will enjoy learning about coronavirus as much as I've enjoyed preparing the information for you guys. And you'll see there's going to be some current events in here, as well as some basic aspects of basic cell biology, things we talked about over the course of the semester, stuff about certain organelles. And we've just finished our chapter in the textbook on viruses. So this is really good timing. So again, I welcome your questions and comments at the end. And so as of this presentation, you know, we're experiencing a pandemic. And, you know, what is the definition of a pandemic, right? We've already talked about this. I wonder if you remember the previous pandemics that we talked about. You can actually speak up if you like. What are a couple of pandemics that come to mind that we've already experienced within memory?
2008.
I think you're thinking of the 2008, 2009. Was it the H1N1, right? The influenza H1N1 was one. And let's see, from April 2009 to April 10th, 2010, there were 60.8 million cases, approximately 274,000 hospitalizations and 12,400 so on deaths. Each of those numbers has a range of uncertainty due to the H1N1 virus. But that was a very mild influenza, and by standards of the flu, right? And so my kids got it, recovered from it. Most people did not die from it, as you can see from those numbers. Millions of cases and tens of thousands of deaths. Another one from the 80s, you guys, you remember? Originated in Africa. Another.
Ebola.
Yeah. Oh, Ebola? Nope. I wasn't thinking of Ebola. Ebola usually does not have pandemics. Pandemic, HIV, you guys. HIV, right? HIV, AIDS, retrovirus. We talked about that in the last chapter. That widely circulated in the 1980s, but actually, interestingly, there's a lot of evidence that it has been in the human population for much longer. And to date, it's estimated that about 30 million people have died from HIV/AIDS since the start of the pandemic in the 80s. And today, living with HIV, living with AIDS, living with HIV, and usually controlling it with medicines, about 35 million people. And there's still a lot of people uninfected, or untreated, I should say. So we're experiencing a pandemic, as you see on the slide. The virus causing it is widely known. It's being closely studied, and it's from a family of viruses known as coronaviruses. And I'm going to focus on that today. This is just a very updated slide from Our World in Data. And you can see the daily new confirmed cases for the United States. And this right here is not good, right? You can see that's not a good situation. Part of that could be due to the weather. Part of it could be due to people not sufficiently taking it seriously enough. Here's another figure. This is the cumulative confirmed deaths worldwide. All right, so over time, just more and more deaths accumulating over time. And I don't know, can anyone think of what kind of curve would match this data? I would say a straight line curve. And in fact, it looks like it's trending up slightly at the end there. And this is right up to date with yesterday's data. Quite concerning. So the death rate for coronavirus is hard to estimate. For the pandemic virus is hard to estimate because you really have to know, well, exactly how many people have been infected, which is a challenge if you don't have a lot of testing, and you have to know, you have to have much more knowledge than often we have available at the time. But it seems like for the last 4 months, the estimates are around 1.5% is the death rate, 1.5%, something on that range, which if you talk about millions of people being affected is a lot of people dying. So that's a sad graph. And so the kind of big lessons of this topic in general, pandemics are not a new thing. We shouldn't be surprised by them. And another fact that we have seen is that it's human nature to act reactively rather than proactively. It's hard to get Congress to, or any governmental body to spend, say, billions of dollars perhaps on preparedness for something that might happen in the future, right? That's just human nature. And so hopefully we will learn some lessons from this and do better in the future. There's some good, this lecture will, I think, end, as you'll see, on a kind of a positive note, but we need to be vigilant still. We're still in the middle of it. So overview, I will be talking about the coronavirus family of viruses, and we'll talk about recent studies on the biology of the coronavirus in question, the kind that are causing disease right now. And I'm not going to spend a lot of time really focusing on the mechanisms of human disease and so on. A lot of that's not fully understood yet, but more focusing on the viral life cycle, similar to some of the things that were in the last chapter. And then we'll end on vaccine prospects and how we make vaccines and some of the new information about vaccines. Great. So some basics about the coronavirus family for you guys. So you remember that viruses are grouped into families, much the same way as other organisms are grouped into families. And we have hierarchies of organization, family, class, order, species, and so on. And I was interested to see that viruses are referred to typically now as being a certain species of virus, which is interesting. And so coronavirus family members are typically infect birds and mammals, but there are others that can be infected as well. But for the most part, that's what they are found in. They're enveloped, which means they bud off from a host cell and they take with them some of the membrane of that cell. And we're going to see some images of these things later. The genome of coronaviruses is a large genome of around 30,000 bases. I don't say base pairs because it's an RNA genome, right? You guys, it's a single-stranded RNA genome. And that single strand is in the same sense as a regular messenger RNA, which we also learned about in this class. So it's ready to be translated into protein immediately upon entry into the cytosol. Isn't that interesting? And you may or may not remember, but there are other viruses that have the antisense strand, and they actually have to recopy their genome oppositely so that they can be translated. And so they have to carry an enzyme with them in the capsid, whereas coronaviruses, they're ready to make all of their proteins, and including the RNA-dependent RNA polymerase that they need to recopy their own genome. That's a lot of information, but it's pretty interesting. I hope you realize. So the appearance of coronavirus comes from this crown-like appearance of the virus. You can see it's sort of, I don't know if crown is the appropriate word, but it did get that name. And as of 2020, there are about 50 of species identified that are well characterized of the coronavirus. And so the genome of the virus is packaged, right, into this virus particle. It has a 5' cap and a 3' poly A tail, just like regular messenger RNAs. But it's really long. It's a really big virus. It's a really big genome for a virus. Okay. Yeah, so here we go. The beauty shot. This picture, have you seen this? You guys have all seen this picture, I'm sure. This is the spiky blob seen around the world. This is a short little news article from the New York Times about the illustrators who created this image. And in the article, they talk about how satisfying it is that people see that and it kind of looks a little scary and makes them take more care of washing their hand, that kind of thing. And so this, we're going to be, I don't know if you can see the these spike proteins. Can anyone tell me what is a noteworthy aspect of the structure that you can see from that illustration? What does it look like to you guys? You see any kind of symmetry there? Threefold symmetry, yes? Would you agree? Yeah, so we're going to talk about the trimer of these spikes. It's all about self-assembly. Viruses are really good at self-assembly, and they can often use smaller numbers of proteins that then can build up this large ball-like structure. And actually, this image has been updated lately. I've noticed on the PBS NewsHour, you can see it's really the same file, but they've enhanced it with these kind of pore-like structures that you can see, right? Interesting. So let's talk about some human coronaviruses. You might know some of this stuff from what you've seen. So discovered in the mid-60s, and it turns out there are 7 known coronaviruses that infect humans. 4 of them cause the common cold, and they have really memorable names: 229E, NL63, OC43, and HKU1, right? I mean, man, I'm not feeling too good today. I think I got a case of the NL63, you know. So fascinating, right? So these viruses are the ones that you and I probably have had them and just give us a cold. And colds are unpleasant. They're not pleasant, but they're not life-threatening. And because we've seen them before, we have some previous immunity. In fact, some of the ideas for why some people don't get as severe disease has to do with maybe that there is some cross-reactivity between some of the proteins of these coronaviruses and the pandemic-causing coronavirus. And so that gives them some protection. So that's a very viable hypothesis. Excuse me. So 3 of these 7 that infect humans cause more serious disease. So we have MERS, Middle East Respiratory Syndrome, 2012 to present. It's still floating around, and it's also called camel virus, or people get it from camels, but it's thought that the camels get sick from maybe bats and then pass it on to humans. 35% death rate, 35%, very virulent. You can see some of the symptoms there, not a pleasant thing to catch. And so this virus is not at all well adapted to humans, but it does infect humans, gets into the body and causes disease. SARS, okay, that sounds familiar, right? That stands for Sudden Acute Respiratory Syndrome. And the original SARS was from 2002 to 2004 in China and other parts of Asia, actually spread around the world to some extent, but it did not become a pandemic the same way that SARS-CoV-2 has. That's the current pandemic virus. Both of these viruses, the SARS viruses, bind to a receptor called ACE2. We'll talk a little bit about that in a bit. It infects bats, civets, which are sort of a cross between a cat and a mink or something. They're cool little animals, and humans. And yeah, so in the world with SARS-CoV-1, the original SARS, there were 8,000 cases and 800 deaths. All right, so that's pretty significant. That's like 10%, right? That's quite a virulent virus as well. And as you remember, the current estimates for SARS-CoV-2 currently is that it kills around 1.5%, somewhere in that range. All right, so let's transition a little bit into the biology of coronavirus. And I'm going to focus on a couple of different papers, which I found to be extremely interesting. Also note in this section some of the techniques that are used to study viruses that are brand new techniques. This is a paper that uses cryo electron tomography, cryo-EM. It's like electron microscopy on frozen, that's where the cryo comes on frozen samples. and they do some tricks where they can get really high, much higher resolution images than in the past, than ever in the past. So they use cryo-EM to study the replication organelles, ROs, in their native host cellular environment. So they infected some cells with a coronavirus, not the SARS-CoV-2, but this other one that's related. All right, and then they looked at the replication process. Right? Are you intrigued? Are you guys intrigued by this? How do they replicate? Well, here is the actual one of many, I'm sure, actual images. And then this is the rendering of that image in colors, identifying different things. The little purple things are ribosomes, right? See all these purple little guys here? They're all, it's the exact same image, right? Shown here and here. And so the green stuff inside are these filaments that they believe are the RNA of the viral RNA. And what's really fascinating is that these are called DMVs, right? It's called a double membrane vesicle. Double membranes. Can you think of another organelle that has a double membrane? That's two bilayers, a double membrane? Of course you can.
Mitochondria.
Oh, mitochondria. There's one. There's one. What about one that's sort of, yeah, that is true. That is true. There's another one I'm thinking of, though. There's another one that comes to mind.
Nucleus.
Yeah, the nucleus. All right. And the nucleus has what in its membrane that are in the double membrane spanning across?
Membrane proteins?
Yep. Membrane proteins. All right. Well, I don't know if you remember this, but all that stuff in the nucleus has to get out. All the messenger RNA has to get out into the cytoplasm. It goes through this structure called the nucleopore complex. Yeah, you remember that now. Okay, so what's interesting is that these organelles also have pore complexes, and that's basically what they're showing in this paper. Check it out. So there's the inner membrane and outer membrane in this replication organelle, double membrane with a pore complex. I'm like, wow. And so these are various images of the kind that they use to do these reconstructions. Truly fascinating. And they compare, so if you look really closely, you can see how the membrane has these structures, and they're sort of like, this is a really close up one here. And there's some stuff coming through the pore. Maybe that's RNA going through, right? And you can see a little dark spot here. Anyway, it's a fascinating paper. And then the authors have this model that they're building based on actual images of what they are seeing, right? So here are the viral double strands and single strands, they believe, inside, right? Here's this pore complex. And these are being extruded out. And then this is a protein of the virus that's coding the genome of the virus. And then those coded genome bits get packaged along with the spike protein, right? And other proteins. The M protein is that one you probably see in the cartoon we saw earlier. Fascinating. We'll talk more about the spike protein in a minute. So just to show you techniques that are used to study coronavirus that are really remarkable and revolutionary. Right on. Okay, so why is this paper important? Here's the closing sentence. It says, although the exact mode of function of this molecular pore remains to be elucidated, it seems to be a key structure in the viral replication cycle that is likely conserved among coronaviruses and thus may offer a coronavirus-specific drug target. Right? Somehow, if you could target that pore complex, which probably is made of multiple proteins, you could maybe mess up the replication cycle of this virus. That would be fascinating. So here's another paper. So back to the spike. Two papers we're going to talk about here about this. So here is the cover of Science. You know, if you're on the cover of Science, it's a big deal. It's better than the cover of the Rolling Stone, I'd say. It's really good. And so you can see this paper is about the flexibility of the hinge region. It like wobbles around. It has a hinge. And so the spike protein covered in protective glycan molecules, which are sugar chains. It's a glycoprotein. It's got sugar chains. On the surface of SARS-CoV-2 is the primary target for the vaccine development. 3 hinges that enable it to be highly flexible and facilitate host cell entry. So every virus has to bind to a target cell and somehow get into the cell. And so that is why we study the surface proteins of viruses, because they are intimately related to the infection cycle. And so how it binds to its target. So the target protein, at least one of them that it binds, is called angiotensin converting enzyme 2, ACE2. And so it's a cell surface protein that's involved in things like blood pressure regulation. It's a protease. And so, yeah, it basically, yeah, so let's see, what did I want to say about this? Yeah, so the binding of this spike protein is what seems to induce the endocytosis. So when it binds to these ACE receptors, ACE2 receptors, it actually gets endocytosed into the cell. Cool. Anytime your spikes are flexible, you're going to have much more of an ability to conform and bind tightly. So here's the second paper, structural analysis of the full-length SARS-CoV spike protein from an advanced vaccine candidate. All right, so this was published on October 20th, where they did cryo-EM, again, cryo-EM studies on Novagen's vaccine material. We're going to talk about vaccines at the end, so we'll get into that at the end. So check out some of the figures on the next slide. We'll see some of these figures, but basically they just made a bunch of images of the vaccine. It's in some cases with different things that are called adjuvants. And so an adjuvant is something that enhances the effectiveness of a vaccine. So here it is. Here's the trimer, right? It looks a lot like the beauty shot or whatever they called it before, right? This is the top view looking down. And you can see how these really nestle together quite tightly. Right? They're really intimately folded together. And then this is actually what the real, if you will, real image looks like. But then they sample thousands and thousands of these in all different orientations, and then they're able to create these high resolution images. That's right down to almost the level of the alpha helix. Can you see an alpha helix right there? Check it out. I mean, you can see the chain. It's amazingly detailed. All right. Vaccine stuff. You ready for vaccine stuff? How many vaccines are in development, would you guess? Oh, you don't have to answer. I'll tell you, I was under the impression from the news that there was, 10, maybe 6, 8. There are actually 48 in clinical evaluation. This is just a screen grab from a WHO summary document that I downloaded, which shows all the various companies. You can see Sanofi Pasteur, all the way from there to Kentucky Bioprocessing, and so there's just so many. All right, and those are 48 in clinical evaluation. So 164 in preclinical studies. 164 in preclinical studies plus 48 in clinical trials. And so what is a vaccine? How is a vaccine developed? So first of all, if you want to design a vaccine, you need to know two things. Like in the end, you need to figure out, is it safe? Is it effective? You've probably heard those terms bandied about on the news. It's safe and effective, safe and effective. Well, safety has to do with side effects, unexpected inflammation, various other things that could potentially be caused by the virus. So they look for that. They look for that in these trials. And then efficacy. Does it work? Does it protect against disease? And so we are just now getting news that some of these early trials are actually completed. On the previous slide, you see that trials go through first phase, which is usually just a small group, a safety phase mainly. They're not looking for efficacy as much as safety. And then maybe another phase 1 trial, slightly bigger. And then phase 3 would be the really big trial with tens of thousands of people enrolled. All right. And so let's just look at some of the different formulations. So the first step in your actual vaccine design is to formulate it, figure out what are you going to put in this vaccine. And the vaccine is designed to trick your immune system into seeing something that looks a lot like the virus. That's a part of the virus. Usually it's a part of the actual virus, but it's not enough of the virus to cause an infection. Sometimes you actually do use a benign virus with just a little piece of the coronavirus added. So you can also use actual coronavirus that's inactivated, just boiled or something, somehow treated, heat treated, or hit with some kind of cross-linking agent to inactivate it so it's no longer infective. A lot of those are from China, I noticed on the chart. And you can go to this link later if you get the file. You can look at that, look at all these different, the whole list. It's a big, long list, like 4 or 5 pages. So another kind of vaccine would be non-replicating viral vector, which is you make a virus, you package it together in such a way that it can't replicate, but it can still infect a cell. AstraZeneca has one like that, among others. Recombinant spike protein, the S protein, that would be a Novavax, for example, has that. Moderna, I think. No, Moderna is mRNA, I believe. You know, I am not going to say that I think I got this right off the chart. So I think this is accurate. So mRNA is the approach by Pfizer and Moderna, which have recently come out with their results. That's why I put it in bold. Why is that a good idea? We'll talk about in a second. Replicating competent viral vector would be a vector that actually can replicate, but is not going to make you sick, including in the case of Merck having it's a VSV virus with a spike protein added in. Virus-like particles are a fascinating thing. It's similar to hepatitis B virus, where you make a protein that can form little like balls that look like viruses that get taken up by immune cells that can be effective. And then the other nucleic acid-based viruses are DNA-based, and there's some of those. Lots of universities are doing that apparently. I think making a DNA-based vaccine is way easier than perhaps a messenger RNA-based vaccine, because for DNA, you can easily purify your DNA and use that as a vaccine. I could do that in my lab easily. To make messenger RNA a little bit more challenging. In fact, I'm not exactly sure 100% how they purify the mRNA, how they actually make that. Whether it's just straight up synthesized, probably not. Probably they purify it from maybe bacteria or something. I don't know. I don't know how they make it. I'm curious about that. So there's still a lot more I can learn as well about how they do these things. All right, so let's see. So step 1, formulate a vaccine. Step 2, design your study. All right, enroll volunteers, and of course there are different phases of the study. So usually you choose healthy people that you don't want to be previously infected, because that would mess up your data, right? And possibly within a particular age range. I don't know if any of you signed up to do a clinical trial for coronavirus. I signed up, but I never got the call. So a lot of it depends on where you're located geographically, where they're doing the trial and that kind of thing. So let's talk about the Pfizer vaccine. So, one more thing before that. So you might think you vaccinate people and then you just give them the virus and see if they fight it off. Well, in some cases that is done. I think for flu that might be done. But definitely not for this, because enough people get seriously ill from it and we don't have a treatment, right? So so that would be called a challenge trial. At the bottom, you can see the asterisk. Some have argued it would be ethical to do a challenge trial with coronavirus, COVID-19. In that case, you would say choose 100 people, or maybe 200 people, but not thousands, just 100, say. Then you give them placebo and vaccine, and then you infect them with the virus, right? And the thing is, you would know the answer to whether the virus is effective way faster, right, than the current method that's being used. And if that could save hundreds of thousands of lives, or tens of thousands of lives, or even 1,000 lives, would that be ethical, right? The question is, would you volunteer for such a thing? In fact, would you volunteer for such a trial, knowing you might have the placebo and they're going to give you the coronavirus, right? Lots of variables to answer that question. I don't expect anyone to answer it, but it's worth thinking about. But it may be a moot point because what we do now that is considered ethical is you don't give people the virus, you give them the vaccine, you give them placebo, and then you just wait. You have to wait for natural infections to happen just in the environment. And one of the reasons why, ironically, we have the results so early is that early graph I showed with the cases going up. There's so many cases around that it hasn't taken that long for there to be over 162 cases, or 170 cases rather, of COVID, right? So they determine in advance how many cases they need to have statistical confidence in the results, right? Obviously 10 wouldn't be enough. Obviously 1,000 would be plenty. Somewhere in between there, they have a confidence in the results, right? And we've been just doing statistical analysis of our fruit fly data, and hopefully you have a feeling now for the importance of statistics in determining precise, I wouldn't say precise, determining defined levels of uncertainty, right? So look at the result. So this is a quote from the Pfizer news release that came out, 2 days ago? Completed phase 3 trial. Analysis of the data indicates a vaccine efficacy rate, I'm gonna read this whole thing, it's so exciting, of 95%. Look at the p-value. P is less than 0.00001. So it's less than a 1 in 10,000 chance that this result would happen by chance, more or less what that means. 95% of participants without prior SARS-CoV-2 infection. The first primary objective analysis based on 170 cases as specified in the study protocol in advance, right? Of which 162 were observed in the placebo group versus 8 cases in the drug trial group, in the vaccine group. You see that, right? Efficacy was consistent across age, gender, race, ethnicity, demographics. The observed efficacy in adults over 65 years of age was over 94%. Amazing, right? And so these results are based on, I think, something on the order of 40,000 people getting to this trial. Right on, excellent. So I have to share this next bit with you guys because it is exciting news. I mean, can we all just say Yahoo, weehoo, yes? Can we dance? Can we do a little victory dance? It's good. It's going to take a while for this to get distributed, but Trevor Noah had a thing to say about this. This is after the preliminary news. Check this out.
But I did promise you the good news too. And people, this is the news we've all been waiting for.
The drug giant Pfizer said yesterday that its vaccine candidate was more than 90% effective in early testing. Dr. Anthony Fauci, the government's top infectious disease expert, called the news extraordinary. He also says there are likely to be other effective vaccines coming soon.
The news from Pfizer was even better than expected.
I would have been very satisfied with a 70% effective vaccine.
According to a Pfizer press release, out of almost 44,000 trial participants, 94 got sick from COVID. 4 weeks after the start of the 2-dose regimen and 7 days after the second dose, protection from symptomatic COVID was greater than 90%.
We were jumping out of the chairs, screaming basically, this is unbelievable, oh my God. We may put an end to this terrible pandemic.
Jumping out of their chairs, screaming. You know something is good news if it turns a laboratory into the Apollo. The COVID protection rate is 90%. And maybe it's because I grew up watching Dexter's Lab, but this scientist's accent makes me want to trust him more because he sounds like a scientist. I'm not gonna lie, if he had the voice of a frat bro, I might be a little hesitant. Dude, this protection rate is like 90%. Pretty much the opposite of when I bang. But this is amazing news. In fact, right now, if you're feeling chills, a little dizzy, lightheaded, that might not be corona. That might be the symptoms of hope.
Oh, I love it. I love it. The symptoms of hope. Although if you lose your sense of smell, he said, you might want to go to the hospital. So there is a lot of hope. But we need to remain vigilant. It's going to take months for these vaccines to be distributed. Thank the Lord for science. Think about 1918, the flu pandemic in 1918. They didn't know that it was caused by a virus. Very little was known. And now we can create these vaccines so quickly, and there's so many of them in the pipeline. Very, very hopeful. So in the future, I believe that the knowledge we learn from all the work that's being done now, we're going to see which of these vaccines tend to work best, It looks like messenger RNA vaccines may be something that we see more of. And it's interesting, if you think about the mechanism, you're putting messenger RNA into cells, right? Right away, you start making proteins in the natural way that a virus would be making its proteins in the cell, right? These are viral proteins being made in the cell. They get processed, inserted into the air, put on the cell surface, depending on what the proteins are, right? Whether they have a transmembrane and so on. But they're going to be processed and presented to T cells, and then potentially also induce antibody immunity very effectively. And so I'm very excited about these mRNA vaccines. I believe this is the first one I've heard of that's been done. So very exciting. Time for your questions, everybody. I'm open to questions. I know you have some good questions. Just chime in.
So is an mRNA vaccine creating viral protein?
Yeah, exactly. So a messenger RNA is the thing that, you get that into a cell and it tracks a ribosome and goes right ahead and starts to translate into protein. Yeah. So messenger RNA is encoding for encoding proteins, right? So remember, the viral genome of coronaviruses is actually like an mRNA molecule itself. But for the vaccine, of course, they encode an mRNA that just encodes like the spike protein, for example, most likely the spike protein, and perhaps another protein or that's conserved and therefore important for function. Yeah. Thanks for that.
I was wondering, you'd mentioned earlier that mRNA would have to be kept at a much lower temperature, which is why DNA was sometimes more effective. How would that affect any of the possible uses down the line?
Right. So that is an important thing to keep in mind, and that is that these RNA vaccines need to be kept at basically like dry ice temperature or liquid nitrogen temperature, really, really cold temperatures. Now, my prediction is that they're going to figure out ways of stabilizing the RNA so that it won't have to be kept at that temperature in the future. I'm sure that's an active area of research. And I believe the Pfizer one has to be kept cold like that, but not all of them do that are RNA vaccines rather. I'm not 100% sure on that last point, but I think that is absolutely one of the downsides of it. Right now, currently, one of the negatives of these RNA vaccines is that they're susceptible to damage. Like the RNA molecule is relatively delicate compared to DNA. It has to do with the extra hydroxyl group. It causes greater reactivity, and it can just, split. And if one mRNA gets cleaved in the middle, boom, it doesn't work. You need to make that entire protein, right? So if the RNA gets damaged, that's a really bad thing for this vaccine. So that's why they want to keep it cold.
As a follow-up, how does that work? I know like when I get vaccines at the doctor, they have them on a little tray that they put in the room until the doctor comes in. Do they have to be that cold at that point too? Like, how does that work?
Yeah, I don't know the exact details. They will warm it up an hour before use that day. It's not like it's going to degrade at room temperature in like 10 minutes or something like that. It takes time. But what you don't want to happen is it to be sitting on a UPS truck or a FedEx or whatever, and just getting overheated, and it, you need to keep it controlled. And in order to get the appropriate response, keeping it at -80, you know it's going to be preserved at that temperature. Yeah, thanks for that.
If it were to be given to someone at the same temperature, at a really cold temperature, would it
Mhm.
cause adverse side effects?
Yeah, you wouldn't be able to deliver it at that temperature. It would be like a rock solid. So no, it definitely has to be thawed and warmed up, and it would probably hurt a little bit if they put it in cold like that. You know, even if it's at refrigerator temperature, that's unpleasant. So it definitely be warmed up to room temperature before delivery. I'm pretty confident of that point. Yeah. It's just for storage purposes and for transport purposes, which, you know, not all facilities are going to have those kind of freezers. So at least the hospitals have them, and that's going to be the primary vaccine group that's going to be vaccinated: doctors, nurses, everyone that works in a hospital, basically.
How are they testing long-term effects of like the coronavirus with the rapid testing that they're doing?
Yeah, so vaccines, so long-term effects of vaccines won't be known until long terms of time go by. So there's always the possibility that you might see an increase in, you know, who knows, like heart disease or something you would never have really expected. But the fact is that usually adverse effects are pretty quick. And most adverse effects are pretty rapid, but there's always the possibility that there could be a long-term effect. But the examples of those such things are very, very rare and very, very uncommon. So most people don't think that is going to be a problem, but they will be looking for that. They will actually look for that. So year out, they'll keep monitoring everybody. There's a very well organized vaccine adverse effect response system called VAERS, which doctors will, you know, report any kind of adverse effects. Keep in mind that when this vaccine has done its job, you know, your mRNA is going to be degraded pretty quickly. It's going to be out of there, proteins produced, and I believe it's a 2-dose regimen. So you have a space of a couple of weeks and then give you a second dose. More proteins are produced and further enhances your immune response. But then it's gone, it's out of your system, and all that remains in your system are, of course, antibodies, which have a half-life. They diminish over time, but also memory cells for both producing antibodies and for memory memory T cells, which is a whole separate arm of the immune system. So these memory cells stick with you for decades, for long periods of time. And even though there are reports of, oh, the immunity wanes and so on, people's antibodies go down. Well, that normally happens naturally. There's a half-life of circulating antibodies. But the thought is that if you get exposed again, then these memory cells kick into gear really quickly and you'll fight it off. You won't get sick.
So would that go for not needing a second dose of the vaccine later on in your life? Because like right now, if you get COVID, they're saying you have like 3 months of having the antibodies and then you can possibly get it again. Would that be the same with the vaccine? Do you know?
For the vaccine, they're recommending usually a booster. Many vaccines, well, any vaccine pretty much will give you a better response with a booster. Certain viruses, by way of their biology, may not elicit as good of an immune response as the actual vaccine, strangely enough. They may have tricks to avoid some kind of immune function. So I believe some of the herpes virus family members like chickenpox are in a way like that. So it might be the case that a natural infection doesn't offer as much protection as a vaccine. So theoretically, that's possible. On the other hand, I would be surprised that someone who recovered from COVID naturally, I would be surprised if they got sick again from it, because a typical virus will generate all kinds of different immune cells that they'll at least diminish your disease so you don't have severe disease again. And there have been some pretty well documented cases of people being reinfected, and it's thought that they had perhaps a milder infection in the first time and then a heavier dose infection the second time. And if you think of how rapidly viruses can replicate, then it's possible that the virus could win out over your B and T cells and sort of temporarily win the race and make you sick again, put you in the bed, put you in on a respirator. But those are kind of rare occurrences. I think the majority of people are going to be protected by a natural infection. Not that they shouldn't get vaccinated. There's always an outside chance it was some other thing that made you sick if you didn't get tested early on, for example. So I've talked to students that, they got sick, they lost their sense of smell, their family got sick, and then they never got a test. They never knew for sure if they had coronavirus. Presumably they did, but still, they can still be vaccinated. Oh, good questions, you guys. This is great. What do you think? Did you learn a few things? Yeah, I mean, I learned a lot putting this together. I appreciate the opportunity. So further questions? Let me show you one thing. I want to show you this other. I got couple supplemental slides. Are there any chats? Someone can chime in on a chat. At the very last slide, actually, I posted. You can't read this, but I'll post this on the LMS for you guys. But these are my Corona Link archives. The first one is way up here, and it goes 3 columns all the way to the very latest ones I put in from October and so on. So some of these are links to podcasts. Some of these are Atlantic articles. Some are just different people's articles over time. So I encourage you to read some of those if you like over the break. So this is an excellent article. This came out in March, in the middle of the sort of first wave of infections. And it illustrates how data can be presented in different ways and give you a whole different impression. So this is the same data, left and right, same exact data. The numbers are the same. The tables of data are the same, but don't they look really different, right? And so the difference is, this is plotted on a linear scale. So 15,000, 30,000, 45,000. So linear scale dates here, all right? And the dates are, this scale is the same on both charts. But this is a logarithmic scale, 10, 100, 1,000, 10,000, 100,000. And so on a logarithmic scale, because the virus replicates in spreads in sort of a logarithmic fashion, 2, 4, 8, 16, that kind of a thing, you end up with a straight line on this graph instead of this logarithmic line, right? It turns it into a straight line. Interesting. And if it's a straight line, that is a bad thing on a log scale. That means that you are still in the exponential phase, right? This is all during those, remember the flatten the curve days, right? Flatten the curve. So you want this curve to sort of flatten out. Anyway, so what you can see is if you were to look at these two countries, you say, wow, Italy is just doing much worse than the US. And this is back in March. But if you look at this curve, you say, oh, Italy is actually curving down. They're slowing they're actually taking steps that are slowing the amount of spread, whereas the United States is actually curving up slightly, which is bad. It's definitely going up versus down. So keep that in mind. There's Our World in Data where you can see things and you can switch back and forth between the log and the linear graph, and it's quite fascinating. Any thoughts on that? It's pretty neat, huh? I know some of you guys like mathematics and appreciate it.
Are you saying that the logarithmic is a better representation of cases or just a different way of representing cases?
Well, if you want to compare the numbers, I think this graph on the left is better. If you're just looking at the raw numbers of cases, right, it's clear that Italy has more cases. But if you're trying to figure out the trend of the rate of change, then the log scale helps you. And actually, you could even do other data manipulations and actually plot the rate of changes, right? That's getting a little into calculus, but that could be plotted as well using this data. But this is actually the exact same data, just plotted by eye. You can see different things in each presentation. So it's not like one is better than the other. They're just different. Thanks for the question. All right, so thank you again, you guys. Excellent questions.